Okay, let me start my presentation. Can you see the screen?
Yes, ma'am.
So did I complete this portion? Instrumental analysis, instrumentation of UV visible spectroscopy. No, right? Hello. Okay. Have you seen my screen? A particular portion of the radiation. For that, filters are used. Those filters are called wavelength selectors. Okay. The sample containers. Since we are dealing with the solutions, okay, the sample containers are very important, okay, to hold the solution. So the sample containers, details about the sample containers, shape of the sample containers, and which material it is made, all those things we are going to discuss here. And detector to detect the signal from the emitted light, okay, and readout devices, okay, printout devices and all these things. Okay, so there are two things are very important here. Two different types of spectrophotometers we are going to discuss here. One is a single and double beam instruments. Okay, what is single beam? What is double beam? Single is only one beam of electromagnetic radiation is used. Double beam, two beams are used. Okay, applications of spectroscopy. Spectrophotometry is more suited for, as I explained you, it is for qualitative as well as quantitative analysis. But this is clear? Hello? Hello?
Ma'am,
Yes, ma'am.
repeat one more time, please.
Yes, ma'am.
Which one? Which one you want?
Application
Hello?
part.
Hello?
Application part.
Application, ma'am.
But your voice is not clear to me.
Application कर दो मैम repeat.
Application, ma'am.
Application we are going to discuss in detail. Okay, that is it is used for qualitative analysis as well as quantitative analysis. Okay, we are going to discuss. Okay, detail. So now we are going to, I am showing the spectrophotometer of single beam. Okay, so this is, let me allow him. Someone is entering. Okay, so instrumentation, this is the source. Okay, the source. Okay, from here the electromagnetic radiations are emitted. Okay, it comes to the wavelength selector, and from the wavelength selector it enters into the aperture. Okay, it is actually aperture makes the wavelengths together as a single beam, and then that goes into the sample cuvette. The sample holder is otherwise called sample cuvette, and then the detector, the beam, the transmitted beam goes to the detector. Okay, this is the basic arrangement of the spectrophotometer. Clear?
Yes, ma'am.
मैम ये sample cuvette क्या होता है?
Sample holder.
Sample holder, okay, ma'am.
Okay. Cuvette is nothing but, so someone's mic is open, I'm getting disturbed. The cuvette is sample holder. Okay. So these are all the features, essential features of spectrophotometer. And source, coming to the source, separated, these are all source, detector and everything. Now we are going to see in detail which type of source is used, what is that wavelength selector and all these things. Okay. So single beam, in a single beam, before that, what is single beam instrument? The light beam follows a single path, okay, from the source. Okay, the beam shows a single path, only one beam, one set of beam is from the source to the monochromator to the sample cell and finally to the detector. Okay. This is a single, only one path, single path it follows, only one path. Okay, that is called a single beam. Okay. So the source, we are going to discuss on the source of light. Okay. The source used in the UV visible spectrophotometer are continuous sources and sources of radiation. So continuous sources emit radiation of all wavelengths within the spectral region. Okay, that is called continuous source. And source of radiation should also be stable and of high intensity. It should have a high intensity. What are the continuous sources we are going to discuss? The continuous sources, visible and near IR radiation. Okay, so there are in ultra UV visible means it has two types of radiations. Okay, one is ultraviolet radiation and visible radiation. Ultraviolet doesn't have any color and visible as a color, right? This you are sure about it, right? Beta?
Yes, ma'am.
Yes, ma'am.
Ultraviolet is a colorless part and visible is a colored part. Okay, so for visible region, the tungsten lamp is used. Okay, the lamp source for the visible light is a tungsten lamp. Okay, so what is the wavelength, visible wavelength? Okay, from 320 to 2500 nanometer. I mean, strictly speaking, the visible region is from here, you can say that around 200. This I will correct it. Okay. So here it is ending up 200 to 400. So 400 to 800. 400 to 800, leaving this near IR part. Okay. So visible region contains 400 to 800 nanometer here. 400 to 800 nanometer. In the case of ultraviolet radiation, deuterium lamp is used. This is from 200 to 400 nanometer. Okay? 200 to 400 nanometer. Is it clear now?
Yes, ma'am.
Yeah, ma'am.
Okay. Next, coming to the wavelength selectors. What is the job of the wavelength selector? The particular wavelength, required wavelength, it will select. For example, here if you see that we need this range, from this to this range, okay, not more than this or less than this, okay. The selection will be made by the filter, wavelength selectors, okay. So initially, the output of the wavelength selector should be the radiation of a single wavelength. Okay, no real wavelength selector is ideal. Okay, so if you see the wavelength selector, it is not strictly, sometimes some defect can happen. This we can discuss later. Okay, so what are the wavelength selectors? It can be simple filters. Okay, simple prisms can be used or monochromator. Okay, the monochromators. Monochromators are generally the gratings, and I will explain this part. Okay, so the filter is a prism. Okay, prism made up of quartz. Okay, quartz or or you can say the glass. Monochromators, if you say that monochromators is a grating, grating is nothing but it is a small plate, a glass plate or a cuvette, quartz plate. Sorry, glass plate or a quartz plate. It has a number of slits. Okay, number of slits, many number of slits. Slits means open. Okay, number of slits. Here, various range of wavelengths can be selected here. Okay, the filter is a simple prism. Prism, you must have studied about the prism. Yes or no? Beta?
Yes, ma'am.
एक स्टूडेंट रिक्वेस्ट भेज रहा है मेरे को ज्वाइन की.
इतने लोग लेट एंट्री है. Just a minute. So the filter wavelength selectors of filters are monochromator. The filter is a single prism. Monochromator is a glass plate or a quartz plate. It has a lot of slits to filter n set of, variety of wavelength range. Okay, here only one particular wavelength range can be selected. Okay, using the prism. Okay, so suppose your solution contains more than absorbing species. Suppose your solution has a two species are there in your solution. For example, your solution has methanol, ethanol. Okay, for example, this is called more than one species. Okay, so in that case, your methanol and ethanol, dono different, it should give 2 peaks, right? Okay, it should give 2 peaks like this. Okay, if suppose only 1 peak hota hai like this, that means some interference is there, with some reaction is there between the species 1 and species 2. That's why it is giving only 1 peak. Okay, that is called interference. Clear? Okay. Suppose you have a mixture ethanol, methanol. So it should give 2 peaks, 2 separate peaks. Beta ye samajh gaye?
Yes, ma'am.
Okay. So in case it is giving only 1 peak, some reaction is going on between these two. That's why you are getting only 1 peak. Okay. That means another substance is interfering. Okay. This is the message. So second species does not interfere in the determination. It should not, because otherwise your absorbance will be different. Absorbance will be different. The concentration will be different, right? According to the Lambert's-Beer's law, you should recall. So now we are going to discuss cuvette, sample holder. For visible and UV spectroscopy, there are two region is there, visible region and UV spectroscopy. When you use a liquid sample, the sample container is called cuvette. So glass, as I told you, the cuvette can be made up of glass or quartz. Glass made cuvettes can be used for the visible region. Okay, the visible region 400 to 800 nanometer. 200 to 400 nanometer if you use, that only quartz made cuvettes should be used. Got it? Beta?
Yes, ma'am.
So now you can understand this is the thickness, 1 centimeter, right? So the cuvette, this is your cuvette, the sample holder. The sample holder has a 2 faces, opaque face and transparent face. The transparent face should face the source. Okay, this gives the thickness of the cuvette. It's shown here at the bottom. There are different types of cuvettes can be used. One is a long path cuvette and 1 centimeter path length cuvette. Okay, these two. According to that, your calculation will vary. The Lambert's-Beer's law will vary. Okay? ये समझ गए? Beta?
Yes, ma'am.
So next, coming to the detector part. Okay, so the detector, so detectors are devices that convert radiant energy into the electrical signal. Whatever the transmitted light, it converts into the electrical signal. Sensitive and fast response should be there, okay, in the detector. And the electrical signal produced by the detector must be directly proportional to the transmitted intensity. As I told you, when the light is transmitted, the transmitted light is converted into electrical signal. This is the job of the detector. What type of the detectors are used in practice? Phototube is one type of a detector. It is a photosensitive cathode is there. How it functions when the transmitted light falls, it goes to the, it is reflected to the from a cathode to anode. It goes as an electron, and the amplifier amplifies. Okay, the signal. Okay, this is how it functions. You don't have to go in detail. You should shortly you should know that how the phototube is used function. Okay, so this is all about the phototube. The second is the photomultiplier tube. The photomultiplier tube is like this. Okay, the electrons are multiplied here from cathode to the anode. Okay, this detection is best compared to the phototube. Okay, so the photomultiplier tubes are used currently in the place of phototube. Phototube is the older version. Okay, better.
Okay, ma'am.
Detectors are 2 detectors, phototube and photomultiplier. Photomultiplier is the most advanced detector. What it does, it converts the transmitted light into the electrical signal. Okay, bus. Then, so now we have to discuss what is a single beam and double beam. Okay, the single beam, as I told you, the source is coming from the source, electromagnetic radiation source. The light is coming. Light through the slit, it goes to the grating. The grating, as I explained you, it is a wavelength filter. Okay, and it filters. You understand now? It has a various slits. Various slits means n number of opening. Okay, so it can filter selectively red, orange, yellow light, or green light, blue light. Okay, it will send, it will be keep on rotating, and it will send only the particular beam. Okay, so it goes to the sample and then the phototube. This is the single beam. Got it, beta? Hello?
Yes, ma'am.
So rotating, it rotates, so it can be rotated like this. And the blank sample, so for example, so this is one, the single beam, this is how it functions, okay? So here, particularly I want to say this sample, okay, suppose you are taking potassium dichromate solution. You have dissolved potassium dichromate solution in water. So before measuring the absorbance of potassium dichromate, you have to check, is there any absorbance in the water? Blank, that is called the reference sample. Okay, since you dissolved in water, you have to first keep in the sample holder the water, record the absorbance, remove this, and then place the potassium dichromate solution. This is how it has been done. Okay, so in the single beam, whenever the single beam spectrophotometer, you have to keep the reference sample and each analysis. You got this point? You have 10 samples, for example, 10 different solution of potassium dichromate. You should have a 10 different blank. For every sample, you should have a blank. That means 20 times your analysis you have to do 20 times. For every sample, one blank. Okay? In the case of the single beam spectrophotometer. Okay, so on the other hand, okay, so if you see here, you can see the arrangement properly. Here, the reference cell and the sample cell. Every time you have to replace. Okay, so this is a single beam. In the case of the double beam. Okay, in the case of the double beam, what is this double beam? Suppose here the source is there. Okay, source you can see that. See this source also you can change it. So UV source or visible source. Okay, you know that two sources are different. Okay, so the electromagnetic radiation is coming here. It falls onto the monochromator and it is converted into a single beam. In the double beam spectroscopy, you have an additional mirror. Like this. Okay, transparent mirror is there. So first, whatever the beam coming onto this, it falls onto the mirror. Okay, this mirror reflects. Reflects light and goes to the another mirror. So it splits the single beam into the two beams. So understood? Beta? Hello. Hello. Hello.
Yes, ma'am.
Yes, ma'am.
So in that, after splitting, one beam goes to the reference cell, another beam goes to the sample cell. Okay. So these two reference as well as sample cells are sample holders are together in the sample, because here two beams are generated. You can analyze reference and sample at the same time. Got the point? So here 10 sample means you need not to do the put the reference every time. Okay, so you can do only 10 times analysis. Got it? On the other hand, single beam, you have to do the 20 times. First, you have to measure the absorbance of the reference sample, and then you will replace with the sample cell. Okay? So, then every beam, whatever transmitted beam goes into the mirror again, it will be reflected and goes back to the detector. Okay, so this is how the double beam function. Okay, double beam is more advanced technique compared to the single beam technique. It reduces the time of analysis. Okay, and also the reference sample, the subtraction, whatever, if you put the absorbance of the reference and sample, it will be subtracted automatically. Okay, so it's automatized, and also it is very simple, it's not time consuming at all. Okay, very fast you can do the analysis. This is the advantage of double beam spectrophotometer. And single beam, double beam, now you understood what is single beam, double beam.
मैडम जो आप कह रहे हो कि इनको रिप्लेस करते हैं रेफरेंस और सैंपल को, वो क्यों करते हैं?
This I explained you that suppose you are having a solution. Okay, let me allow him. Reference and क्यों करते हैं? Suppose you are dissolving in water, okay, potassium dichromate. Your water can contain some impurities. Do you agree or not?
Yes, ma'am.
Yes, ma'am.
Okay. So the interference of that impurity should not interfere in your absorbance of your experimental sample. Fine. That's why you subtract. Suppose your absorbance of your sample is 20, actual absorbance. So if you don't subtract the absorbance of the water, it will read as a 25. Okay. If you calculate the concentration of your sample based on this, you will end up with the erratic reading. Agree? So the extra 5, if you measure the absorbance of water separately as a 5, and if you subtract it, okay, 25 minus 5, you will get only 20 actual absorbance of your sample. Better understand?
I'm एक बार repeat कर दो.
Better, here reference sample you are taking, you should take a water. Here you are taking in the potassium dichromate solution. 1 minute, I'll project it. See, potassium dichromate solution here. Okay, so water, suppose if you put water here, water will have absorbance. Water, strictly your solvent should read zero absorbance, but it is not possible. It may have some impurities. Due to that impurities, it may show some absorbance. Okay, 5, 2, something like that. Okay, your sample has a absorbance. Okay, so actually your sample means you have dissolved it in a water. Okay, sample has separate absorbance. So actual absorbance of your sample, you can get it by subtraction of the absorbance of your water sample. Okay, that is called reference. Got it?
Yes, ma'am.
So single beam, double beam, you understood what is single beam, what is double beam. Okay, single beam, the mirror is not there. A double beam mein mirror hai, so it will split the beam, whatever is coming out from the monochromator, into the two beams, two parts. Okay, nowadays
डिटेक्टर पे तो वो एक साथ जा रही है ना दोनों इंटेंसिटी, तो फिर डिफरेंशिएट कैसे करेंगे?
हेलो बेटा, आई एम नॉट गेटिंग यू.
मतलब इस पर डिटेक्टर पर तो एक साथ जा रही है ना दोनों इसकी इंटेंसिटी,
यस
रेफरेंस सैंपल की, तो फिर वो अलग अलग कैसे करेंगे सबट्रैक्ट करने के लिए?
अलग अलग सी दैट इज ऑटोमेटिक, दैट प्रोग्राम्ड. दिस होल